Welcome to this introduction to the Point Cross Xbiom Workbench for clinical and translational research insights. In this presentation we will show you how various modules of Xbiom work to support the entire range of activities from accessing disparate data sources and standardizing, and semantically enriching, the data for search, analysis, and generating insights and metadata, to visualizing and repurposing. Let us begin with a short introduction to Point Cross. Point Cross Life Sciences has been serving the biopharma industry since 2009 with technology solutions to serve research analytical needs, as well as regulatory workflows. Our solutions span discovery, non-clinical drug safety, clinical, and translational research. Xbiom is our solutions platform that includes various optional modules to serve the immediate needs of our clients while providing for growth into other adjacent areas of research or operations. In this presentation we will cover all the functional modules that are needed for clinical, translational or biomarker research. Point Cross serves many of the major biopharma companies including Roche, Sanofi, Bayer, Ipsen, Eisai and about 25 others across the US, Europe and Japan. We are a platinum member of CDISC and a contributor to various data standards. We have been a FDA contractor since 2011 and we provide risk modeling and simulation technology to the Center for Tobacco Products at the FDA. Point Cross is located in multiple facilities in US, France, and Bangalore, India. Biopharma companies collect a large variety, and a large amount, of data from various studies and assays conducted at internal and external CROs or labs. Xbiom can ingest and normalize data from non-clinical studies and clinical trials. The data may include subject health history, their ECRF data from EDC systems in their native operational data models with all their clinical findings, various PK/DMPK studies, molecular omics biomarkers covering genomics, transcriptomics, proteomics and metabolomics, immunohistochemistry and flow cytometry data. Xbiom can support functional biomarker data obtained from various remote monitoring devices and POCTs. Integration to clients' biobanking facilities provides scientists a real-time view of the availability and viability of biosamples for additional sequencing. Often, patient data may be limited to specified research as allowed by informed consent. All research data is dynamically filtered to include data that a user is permitted to see based on their roles and therapeutic research area. Xbiom ingests these disparate data so that various analysis tools can be applied by researchers or data scientists and use the onboard immersive graphics object we refer to as IGO, or one of the many standard third-party tools to visualize the data or results of analysis. Xbiom consists of a number of standard modules that can be turned on or off depending on the solution needed by the client. The modules across the center are essential good housekeeping modules that brings the raw, uncurated, and non-standard data into a searchable, accessible facility for researchers to do their science on the data rather than wrangle with untamed data. Xbiom's smart transformation connects to dispersed data sources within and outside the enterprise to source data that is useful for analysis and research. This module also includes management and governance facilities for data models, standards, metadata, and terminologies with ontology management and semantic enrichment. The core ontology platform is the module that actually holds all the data, metadata and provides the essential user management and security functions. It connects with the enterprise network and the runtime facilities for scheduling and crawling data stores or running various tethered or phase-gated workflows. The eData Validator is a rules-driven engine to test data models of various data sets against established conformance rules of CDISC, FDA or PMDA regulatory agencies. The validator is also important when establishing a data exchange format with external labs so that labor for curating and quality checking can be reduced or eliminated. Although the diagram lists modules non-clinical study data integration for toxicology and drug safety research, and modules for regulatory preparation and analysis for submissions to regulatory agencies, the focus of this presentation is the module on top left that refers to the workbench for translational and biomarker research. Xbiom is available on a software as a service basis installed in the AWS Cloud Services. It can be installed on-premise as well. The dedicated, hosted SaaS offering can be stood up in a matter of days, and the subscription model makes it an unbeatable value given the various updates and availability of functions that meet the growing and evolving needs of the biotechnology industry. This slide shows one variation on how the Xbiom clinical and translational workbench can be configured. In this representation, the Insights module gets clinical data from the clinical trials sources that are held in GXP-validated servers. This can be replaced by a connection to the client company's ClinOps servers. One of the prevalent problems that we come across is that biomarker and translational research departments find it very difficult to get clinical data from the ClinOps divisions. The difficulty has to do with the risks of sensitive patient data getting out of the controlled environment within ClinOps. A read-only integration as shown here is an important and simple solution because it allows for control over what data or which studies can be permitted for use within Xbiom without letting that data sit in an uncontrolled server or data store. Not all the clinical data collected is needed for research, only a small subset is required. And, this is established in the Smart Transformation module to control what can be ingested. Beyond the clinical trial data are the various assays that are conducted by researchers or to support submissions to the regulatory agencies. The Smart Transformation module reads these data sets, and based on the training done on the AI machine learning algorithms, recommendations are provided on how the data should be mapped to the target data model. Where the incoming data meets a previously established standard in the standards library, the conversion is automatic and only a validation is done, automatically. Once the data is ingested after standardization and semantic enrichment, it is indexed and available for search for stratified cohorts and extraction of analysis data sets. For those who prefer to work with SDTM standards, a statistical computing environment, the SCE, is provided so that validated legacy scripts can be reused. The search engine accelerates the generation of analysis data sets without having to process the entire trial source data sets. Xbiom is compatible with OData standards that make it possible to exchange data or interact with Xbiom from any third-party solution for analysis or visualization. The use of interactive Jupyter notebooks and running R or Python scripts along with the use of TensorFlow machine learning tools from Google make this a true researcher's workbench. Xbiom also has a built-in configurable data visualization toolkit, the IGO, a immersive graphics object built on D3. With the OData interface, Xbiom is frequently accessed by other enterprise applications to avail of the various functions and facilities embedded within. For biopharma companies engaged in translational and biomarker research, dealing with a myriad of standards and data models with the ability to transform data from any source format to any selected target model is a necessity. The same is true for managing terminologies and various code lists and maintaining the ontology for the company. Xbiom provides facilities for the management and governance of standards. Whether they are de jure, de facto, or a client's custom data models, these standards are versioned, and maintained in an accessible library where specialists can maintain and create new standards model as needed. Point Cross provides frequent updates that let clients stay abreast with the prevailing de jure standards such as CDISC, SDTM, ADAM, and SEN standards. We also provide de facto standards for various omics assays that are commonly seen in the industry. The data collected for translational research includes in vivo trail data, in vitro and ex vivo assays containing disparate terminology. Molecular biomarkers in omics assays from external labs have lab-specific notations for genome variants and gene names. Clinical data contain adverse events and diagnostic terms that must all be semantically normalized, or enriched. This means that just like standard data models, various code lists must be maintained and all concepts that may have terms in one or more codelists must be related and mapped so that every concept has a unique invariant term associated with it with the ability for the ontology engine to map it to any other term dynamically. A simple practical use case is when a scientist searches for data for a particular test name. The search is conducted by Xbiom using the invariant globally constant term in the ontology engine, thereby finding data across the entire store across all assays and studies irrespective of how it was originally reported. The runtime aspect of smart curation is the ability to take the source data and preparing it for ingestion, first by transforming its data model to the target model with minimal, if any, intervention, followed by normalizing all the terms using the ontology engine. A quality control, or QC dashboard provides the controls for monitoring the completeness and quality of the transformation. Xbiom provides all these facilities pre-integrated because these are essential functions to make the data useful for researchers. Source data is transformed into standardized and semantically normalized target data sets. Xbiom does this with two different target models. SDM, or Standard Data Model, is a selectable data model from the standards library. An example may be a specific version of SDTM for clinical trial data such as the ECRFs. This SDM can differ based on the business needs of the users. One typical use case is to generate standardized data that can be operated on legacy analysis tools, while another reason is to be able to exchange data readily with partners and collaborators. The other target model is the UDM, or Universal Data Model. The UDM is flat, simplified and meant for machine readability and search indexing, or for generation of linked object data models such as graph models. SDMs are not very useful for data integration. The common standards such as SDTM, ADAM and SEND are meant for exchanging data among parties. Data integration was not a consideration when these standards were developed. To integrate data, it is essential that all data, however it was received, is in an invariant and simple model that allows a machine to consume it and regenerate the data in any form or standard that may be of use in the future. Therefore, the UDM supports regeneration of data in any of the appropriate standards of that time. Xbiom is almost always installed with integration to internal or external data sources. These include integration for read-only access to clinical trial data, integration to biomarker data stores such as iRODS, and external biobanks for biosample availability. There is no limit to external or internal data stores and sources that Xbiom can be integrated. Labs can upload data from assays directly to the VDR, or virtual data room in Xbiom, where it gets automatically picked up for transformation. Xbiom also integrates to various registries in the cloud. These omics registries for genomics, transcriptomics, proteomics, metabolomics include known variants of biomarkers and references with annotations to help researchers. Xbiom will periodically crawl these registries to update its ontology engine. Xbiom also connects to various standards and terminology registries. Point Cross provides updates for standards and terminology periodically if the Smart Transformation option is included in the subscription. The value of integrating disparate studies, clinical trial data and molecular biomarker data is to be able to find cohorts that meet very carefully defined criteria so that the data associated with these stratified cohorts can be compared differentially. These may show important trends longitudinally that may point to changes in endpoints due to disease progression or dose-dependent efficacy. Discovering differentially expressed genes or the presence or absence of certain gene variants can be important clues to the translational scientists. Many of the biologics-driven therapies are precisely targeted to specific pathways and receptors. In some cases these therapies can be very effective only for some genotypes and not others. These point to companion diagnostics to find and select the most likely exceptional responders that will react positively. The search interface is split into separate areas that include study or assay attributes and summaries, patient health history and baseline data, subject clinical parameters and endpoints, molecular biomarkers, and biosample availability data if this is available. Recently we added SDTM as another search tab because of the interest from biostatisticians and programmers who are used to ADAM and SDTM data variables. If non-clinical studies insight modules were included, there would a couple of tabs for those in vivo studies. Similarly, if discovery assays are included, added additional tabs may be needed. Users will typically set up their search criteria limiting their search within a candidate molecule or sets of studies or open it up across any and all studies in the system. These search criteria can be saved and reused, and they may be published to the larger community for collaboration. The search results can be saved. The search criteria from various tabs and from within the tabs can be combined using Boolean logic to create very powerful search query masks. In each tab, the left side of the search screen will show all the data that was collected across all the studies. The user is provided instantaneous cues on how many subjects will be found as the search criteria is built up. For quantitative data, a histogram and the limits on the data available for that parameter is cued to the user. For example, consider a scientist who wants to find cohorts treated with ipilimumab, and separately, for another candidate compound, PCXX, to discover changes in the genes and expression levels involved in pathways and the immune levels in the colorectal cancer tumor tissue environment. Xbiom's query masks allow scientists, without help from programmers, to set up and find these cohorts from metadata and clinical trial data across the entire repository of all studies held in Xbiom. Search is set up on study summary, subject's health history or baseline at recruitment, their clinical test parameters, all omics molecular biomarkers and even the availability of biosamples for additional sequencing or assays. Multiple tabs are available for each category of the search queries, and the search queries can be tiered within each category. Study attributes that define the study types, treatment drug, and therapeutic areas are available in these tabs. The researcher may look for subjects with specific endpoints, based on surrogate measurements, or as calculated from analysis. The molecular biomarker search allows users to search for specific variants in the various genomic components, such as transcriptomics, proteomics or metabolomics. In this scenario, patients treated with ipilimumab or PCXX compound are selected from the study summary level by searching for disease, indication and candidate compound from the left side bar, and further selection done for the ethnic group. Molecular biomarker for cell type and RNA data are then chosen for the advanced search. As the query mask is developed further, the scientist can filter data sets on the cell type in flow cytometry to choose the CD4+ T cells and CD8+ T cells with a minimal observed percentage value. RNA-seq data for suspected gene sets that potentially participate in the colorectal cancer tumor are then selected from the gene list. This search can now generate analysis data ready for differential expression levels of the two treatments, along with the T-cell percentages at the tumor environment. Obviously, the search could also be done with clinically discovered endpoints or lab findings with the entire omics results from the selected cohorts differentially analyzed separately. The gene enrichment can be done with tools such as DE Browser. In this slide, the differential gene expressions can be plotted using the DE Browser with tools such as DE-seq2 and visualize it in Volcano or MA plots using a simple R-Shiny app in Xbiom. Another use case is gene enrichment of the cohort, or subjects within the cohort, to then refer to the KEGG pathway to view implicated pathways affected by the differentially expressed genes. Pathway diagrams can be dynamically pulled down from the KEGG registries. DE genes may also be enriched through gene ontology database and disease ontology to further understand the change in expression levels of genes due to the treatment molecule. Xbiom has a very rich, and configurable, data visualization facility called an IGO, or Immersive Graphics Object. IGO allow researchers to view data, search results or analysis results. Quantitative or qualitative results of biomarkers and clinical data can be plotted for interactive analysis and discovery of insights. This concludes our brief, high-level, introduction to the clinical workbench for translational and biomarker research. Please take a moment to note down our contact information, and please do reach out to us for more information or to get one of our scientists answer questions you may have. Point Cross has gone to great lengths to make Xbiom easy to try with a pilot using your own data. Xbiom is almost instantly available as a dedicated instance for your company hosted on the Amazon AWS cloud. We connect to your company's network through a virtual private network for single sign-on, and to connect to your various data sources. If your company already has an Amazon AWS account, or you wish to use your own on-premise for installation, Xbiom can be easily installed by your IT organization. Once stood up, Point Cross can help in standardizing and normalizing data sets from your data lake or disparate sources to ingest using the Smart Transformation module of Xbiom. Working with many major biopharma companies, we have done this in a matter of weeks. Reach out to learn more. Thank you for watching this presentation.